Okay, thank you. Thank you, Nancy, for inviting me. So I was asked to talk about what we're doing in Norwich with related to ME research. And as you can see from the title of the talk, we're interested in the gut-brain axis and how things that happen in the gut might influence behaviour and some neuropathologies, in particular those that affect ME patients. So this is sort of where we began, and basically looking for clues as to what the underlying cause of ME might be. So there's quite strong evidence that it's immune related, and Joe Cambridge has already mentioned this. So immune related symptoms are very common. Often episodes of disease are associated with or occur quite soon after an infection, particularly viral infection. Patients, as Joe has already said, have altered populations of B cells, and the B lymphocyte depletion therapy, rituximab, is proving quite effective. And what's interesting to us is that more than 90% of patients usually have some GI disturbance as part of their symptoms. So the question really is then, is ME an autoimmune disease, and does it originate in the gut? That's the sort of premise of a lot of the research we do. This is our hypothesis. So ME patients may have a leaky gut, and I'll explain what a leaky gut is in a minute, that leads to infection by resident microbes. And this is a trigger that causes autoimmune disease. And there's a precedent for this, which is inflammatory bowel disease, so Crohn's disease, ulcerative colitis. They're associated with an aberrant immune response to members of their gut microbes. And we're also interested in sort of applying some of this information that we gain, this basic science, to therapy. And there's a possibility that we may at some point be developing a trial or a treatment based on rituximab. And also we're very interested in using alternate types of therapy to change the population of microbes that live in the guts. Of course, bacteria therapy, and a variety of ways in which we can manipulate gut microbes to improve health. Okay, and these are the hospitals. Norfolk and Norwich University Hospital, which is next to where we are. Community care, we're increasingly trying to interact with general practitioners in practices across Norfolk and Suffolk. And Epsom and St Helier Hospital is where we recruit most of our patients from. So, Hippocrates, many, many years ago, I think probably got it right, in that all diseases begin in the gut. And really what's key is keeping what's in the gut outside the gut and away from the body. And when the gut barrier becomes leaky, or the integrity of the gut barrier is damaged, then this can cause lots of disturbances in a whole variety of organ systems that not just affect the gut, but other more distant organs as well, including the brain. And these are some of the conditions, disorders that have been linked to or associated with a leaky gut. Now, what can cause a leaky gut? Well, there are a variety of things. Some of the obvious ones include infections, diet. Things in the diet can influence the leaky gut also. Stress, hormonal changes, all of these things can impact on the integrity of the gut. They cause the barrier to become less tight, so leaky, and this allows microbes and microbial products to get into the bloodstream, where they can then cause inflammation, which can spread throughout the body. So the important thing here is to remember the gut is not just an empty tube, it's actually packed full of microbes. And these are examples of some microbes, and they do live in close association with the lining of our gut. So there's intimate interactions between our microbes in the gut and the rest of our body. So there's a direct communication between microbes and ourselves. And what we now know, or we're beginning to understand, is that the microbes are playing an important role in whether or not we remain healthy or we are prone to developing various disorders. And so there's a dynamic interaction between our genes, our genetic makeup, our gut microbes, which are strongly influenced by our genetic makeup, and then environmental factors, and in particular lifestyle factors. And it's these three factors which come together, primarily focused in the gut, that then can change populations of microbes, so-called dysbiosis, which can lead to some microbes gaining access to the body and causing inflammation. And these diseases are now commonly associated with changes in the behavior or the makeup of microbes in the gut. So the question is, do we say the same thing in ME? And this is really what we're sort of interested in looking at. Okay, so in particular, we're interested in what's called the dark matter of our gut microbes. So, as I said, most of the information that we know about our microbes concerns the bacteria that live in our gut. And there are some estimates that there are maybe 10 times as many microbes as there are cells in the body in some cases. But what's important is there are also a population of viruses that live in the gut as well, and this is called the virome. Okay, and there are 10 to 20 virus particles for every single bacterial cell. So again, viruses are by far the most abundant species that live in the body, and in fact in the biosphere as well. Viruses far outnumber any other species. So some fundamental questions in order if we're going to address and look at these microbes, these viruses in terms of ME. What is the virome? Why is it important? How the viruses change, and how may that impact on health? And is it important in ME? So the viruses we're particularly interested in are a population that actually live inside bacteria. And these are called bacteriophages, derived from the Greek to devour. And there are two predominant types, which I've shown here. These are the Cordiovirales. They're very characteristic. They have a head shape and then a tail, and almost like a syringe. And then we have these sort of more spherical types called the Microvirales. These are the two most prominent types of viruses that live in the gut. And you can see this is a single bacterial cell here, and you're going to see how many viruses are actually entering or exiting from the bacterial cell. And what's an important point here is that these viruses are very specific. They will only invade a certain type or species or strain of bacteria. They're exquisitely specific. And they've been known for a long time, so we've known since about the 1920s about bacteriophages, and they've actually been used quite extensively to treat bacterial wound infections. So they were used as therapies to treat American and Russian soldiers during the Second World War. And there are some former countries of the USSR that are still using phage as a therapy to treat bacterial infections. And in the US, since 2006, they're actually used to destroy bacteria that may be contaminating foods, like meat and cheese, for example. So phages are commonly used to kill bacteria, as we consider them to be infectious bacteria. Okay. So there are three ways in which these phages can change the behavior of microbes. They can hide inside. This is called lysogeny. So in response to starvation or administering of antibiotics to an individual, the phage can sense that as a threat to their survival, which means they'll then invade a bacterial cell to basically escape from the stimulus that's been delivered. They can actually then become lytic, so they can actually kill the bacterial cell that they live in. And again, this can be in response to different types of stress, so change in the diet, drugs, etc., or nutrient starvation will cause them to move into a lytic cycle. And this destruction of bacteria can cause an inflammatory response as well in the body. And then the third one is far more subtle, in that they'll just transfer certain genes from their own genome into bacteria. So instead of incorporating their entire genome into a bacterial cell, they'll just put individual genes in. And this is interesting because some of these genes are actually related to antibiotic resistance. They can allow bacteria to survive in the presence of antibiotics. And the survival of these bacteria is obviously an advantage to the viruses, because they can then infect them. They have a reservoir of bacteria to infect at some later point. And they can also, bacteria can gain a metabolic advantage. They can outgrow competitors through acquiring some of these virus genes. So these are three common mechanisms by which viruses can change the bacteria that are in the gut. And we now know the virome, this is the very latest study, they're just over a year old now, looking at the virome in patients with inflammatory bowel disease compared to normal healthy controls. And what they showed is basically this type of bacteriophage by far outnumbers this type. This is the Cordiovirales. And this seems to be specific to patients with Crohn's disease and ulcerative colitis. And a very recent study by one of our collaborators in America at Cornell University has shown that actually bacterial populations in ME patients are diverse, change in their diversity as well in the disease. So these viruses, by outgrowing or outcompeting other viruses, can obviously induce changes in bacterial populations that can make them less diverse or more diverse. And ME patients have this similar shift in diversity seen in inflammatory bowel disease patients. So this is the beginning of our study, and this is our gut viruses and ME study. Involves, as you can see, a whole team of players here from not only the University of East Anglia and IFR, but also from hospitals where we're recruiting our patients, Imperial College, providing important bioinformatics help, and also from Uppsala University, where we're looking at metabolomics. And basically, we're collecting stool samples, we're then isolating the virus particles from the stool, and then we can sequence them to identify which viruses are present. And this is what we've shown so far. So what we've been able to do is to determine that ME patients have a particular profile of viruses in their gut, and this is different to individuals and normal healthy controls. And these are some of the prominent types of viruses. You see, again, it's these same Cordioviralis type viruses which predominate in inflammatory bowel disease patients. We're seeing somewhat similar pattern in ME patients. And if you remember, I said each virus is specific for a certain type of bacteria. Based on identifying these, we can also identify the bacteria down here that they actually invade and can infect and maybe even kill. So by looking at the viruses, we can actually look at how they may be changing bacterial populations and in turn then impacting on health or disease in these patients. So these were from a group of patients, or this analysis from a group of patients with mild to moderate ME. And so what we're doing now is we're looking at what happens with these patients as they progress. We're also starting to collect samples from severely affected patients as well. We're having to develop some tools to do viromics analysis. This is a relatively new discipline. And the question we're asking is, do these phages that we've identified in ME, are they responsible for killing bacteria? Are they responsible for the dysbiosis that we've seen in ME patients? And can these viruses actually cause or explain diseases? So we're interested in looking at the functional properties of some of these viruses. And these are two PhD students in my group that are working on this project. And then the other side of this is, do these viruses some way change the immune system? So now the immune system starts to attack these microbes, the bacteria or the viruses, as seen in inflammatory bowel disease. So we're testing the immune response of ME patients to see if they have reactivity to their own gut viruses and bacteria. Again, and the two PhD students working on this as well. And so we're particularly interested in antibodies that get secreted into the gut. So these are IgA antibodies. And we produce far more IgA on a daily basis than any other type of antibody. So there's vast amounts of IgA are secreted into the gut. They play an important role in binding to and eliminating toxins, stopping them from gaining entry into the body. And so in a leaky gut, we would expect that there would be an influx of microbes or microbial products in which the immune system can react to. This stimulates IgA production, and we can detect this by the presence of bacteria in the gut which are actually coated with these IgA antibodies. And in inflammatory bowel disease, IgA coating has been shown to identify the bacteria that actually cause the disease. So we've started to look at this in ME patients. These are severely affected ME patients, so we're going into their homes and collecting samples from them because obviously they can't go into hospital. And this is just very early, this is just a small group of patients, and you can see that they're, in comparison to the same household healthy controls, these are generally carers or parents of the affected individuals. The patients with severe ME tend to have more of their gut bacteria coated with IgA. And this mirrors what we see in inflammatory bowel disease, both the Crohn's disease and ulcerative colitis. There's more IgA being produced, which is coating more bacteria in these patients. So there's a stimulation of IgA production in these patients. So obviously we're interested in knowing what these antibodies are reacting to in these patients, and we're right now in the process of trying to isolate the bacteria that have been coated by IgA to see if they may be able to cause disease in other experimental systems. So there's just a couple of things I want to finish off with. So I was asked to talk about how we're trying to sort of link together research groups across Europe to try and sort of coordinate research activity. And so I'm going to talk about a couple of things that are happening. One is happening very locally in Norwich. So this is an £80 million investment in a new building which is going up on the Norwich Research Park called the Quadram Institute. And the focus of this is going to be on gut food microbiome research. And we're going to put ME research as a central component of the research activity within this brand new institute. This is how the institute currently looks. So it's almost the external components of the institute are already virtually in place now, and we're expecting it to become operational about this time next year. And so this is a summary of the sort of types of research we'll be doing in the new institute. Again, it's food, interacting with the gut, and interacting with gut microbes, and how this is important in early life events to ensure that newborns acquire the right types of bacteria to give them lifelong health. How food is digested, how this influences central metabolism, glucose metabolism. Microbiome host crosstalk, and in particular how microbes in the gut communicate with the brain and how this affects behavior. Intestinal inflammation, how changes in these interactions can lead to intestinal inflammation. How we can use bacteriotherapy to treat abnormalities in gut microbes. And we're hoping that the ME studies will fit very well into this overarching research strategy of the Institute. So that's happening in Norwich, very exciting development. These are two things that are happening further afield. So these are two new European initiatives, EuroMe, which is now an EU-funded program, and the idea is to network people across Europe that are working on ME to form groups, collaborations, and Emerge is to actually advance the ME research agenda across Europe. So this is EuroMe. So it's a €6 million investment, involves various groups across Europe, including here in Belgium, and these are some of the things that they are trying to develop. As you see, it's really bringing people together to standardize approaches, identify cohorts of patients across Europe which can be used for research studies. And then Emerge is really sort of building on this in a way to really identify the research agenda and the priorities. And the flags here show the groups. You see, it's not just Europe. We do have members or interest from Australia and the USA. And these are sort of three work packages, if you like, three areas of research that we think we should prioritize. First one is obviously diagnosis and identifying patients of subcategories of ME. Biomarkers, we've already heard lots of that already. And then sample standardization. And that's particularly important if we're working with stool samples, is making sure they're handled, analyzed, processed, stored in the same way to ensure their integrity. All this will then become available for doing directed research projects across Europe. And so this just really summarizes that it's these two organizations trying to work together. Emerge will establish the research agenda and priorities. EuroMe is establishing networks of research and stakeholders which can then work together on this research agenda. So the two really are aimed at working in closely. And also we're having some influence now worldwide. This is a very interesting development some of you may have heard of. This is from America and the National Institutes of Health. Dr. Vicky Whitmore, she's established a working group to try and coordinate research activity in the US. And they're funding a program here, and these are the goals that they've set for the next year. And you see one of these is to foster and develop international collaborations. And so both Vicky Whitmore and a senior member from the CDC, Centre for Disease Control, will be coming over to the Invested ME Research Conference in June. So we really are trying to join up with the Americans here. And interestingly, several Emerge members are taking part in consultative discussions with members of the NIH, particularly on the area of biomarkers and immune markers. So we are slowly starting to coordinate activity in ME research, not only in Norwich, but further through Europe, and hopefully with the Americans as well. So I think obviously Americans will be able to invest more money, but there's an opportunity for European researchers to get really involved and to get benefit from these funding programs in the States. so I'll stop there. Thank you very much.
Thank you, Dr.